On December 8, 2023, the morning sun illuminates a hospital room in Nashville, Tennessee, setting the stage for a historic medical breakthrough. Victoria Gray sits on the edge of her bed, waiting anxiously to learn if the experimental gene-editing treatment has actually worked. For 33 long years, she endured a level of physical pain that most people cannot imagine, struggling through a life defined by suffering. She battled sickle cell disease, where her own red blood cells twisted into sharp crescents, ruthlessly attacking her body from the inside out. Every single health crisis felt exactly like shards of broken glass were flowing through her veins, causing agony that never seemed to end. No cure existed for her condition, only endless pain management, stacks of medical bills, and a life defined by waiting for relief. That changed when scientists decided to attempt something daring, a procedure that had never been successfully performed. They edited her DNA while she was alive, using a revolutionary tool to cut the very code that defined her existence. They changed the specific genetic code that made her sick, replacing the broken sequence with a corrected version to fix her permanently. Now, exactly 3 years later, the doctor opens the folder containing her results, while Victoria watches his face with held breath. The report confirms she has had 0 sickle cell crises, not a single one, in the 3 years since her treatment began. The disease that tortured her for 33 years was gone, leading to an emotional embrace between the patient and her relieved doctor. This is not science fiction. This actually happened, and it is about to happen for millions of other people around the world. This video reveals exactly how scientists finally learned to rewrite the code of life, changing the future of human medicine forever. We will explore why the next 10 years of medicine will completely change everything you think you know about treating human disease. To understand why Victoria's cure is so revolutionary, you first need to understand what DNA actually is and how it functions. I promise to make this simple for you, so you do not need a biology degree to understand this incredible breakthrough. Think of your body as a massive city, a complex metropolis that is constructed from over 37 trillion individual building blocks. Each building is a cell, and each cell needs a specific set of instructions to function correctly within this massive biological city. That instruction manual is your DNA, providing the essential blueprint that tells every cell exactly what it needs to do to survive. DNA is a language written in just 4 letters, A, T, C, and G, which arrange themselves into specific genetic sequences. It is 3 billion letters long, forming the complete instruction manual for building the unique person that is known as you. If you printed it out, the code is so long that it would fill 200 phone books stacked floor to ceiling. Here is the incredible part. Every single cell in your body contains a complete copy of this massive 3 billion letter genetic manual. Most of this code works perfectly, but sometimes there is a typo, a small error that disrupts the instructions for the cell. Just one single wrong letter out of 3 billion total letters is exactly what gave Victoria the debilitating sickle cell disease. One tiny typo resulted in 33 years of suffering, showing how fragile the balance of our genetic code really is. For decades, scientists dreamed of fixing these typos, but DNA was too small, too complex, and too protected to be accessed. It was like trying to fix a typo in a book that was locked inside a safe within an impenetrable building. Everything changed in 2012 when 2 scientists discovered something that bacteria had known and used for billions of years of evolution. Jennifer Doudna and Emmanuelle Charpentier are the brilliant women who unlocked the secret, changing the course of scientific history for everyone. They found that bacteria actually have their own immune system, designed specifically to defend themselves against attacks from dangerous invading viruses. When a virus attacks, the bacteria cuts out a piece of the virus's DNA and saves it within its own genetic code. It is like keeping a mugshot of every criminal that ever attacked you, creating a permanent database of known threats. If that specific virus ever returns, the bacteria recognizes it instantly and destroys it using the stored genetic information as a guide. The cutting tool is a protein called Cas9, which acts like molecular scissors to cut DNA with incredible speed and precision. Doudna and Charpentier realized something extraordinary, a moment of insight that would eventually lead them to win the Nobel Prize in chemistry. What if we could reprogram these scissors, telling them to cut anywhere we want, instead of just targeting specific viral DNA? We could target human DNA to fix the typos that cause disease, rewriting the errors that lead to suffering and death. In 2012, they proved it worked, publishing their findings and ensuring that the world of science would never be the same again. Discovering the tool was just the beginning, because making it work safely in humans was a challenge that took years to solve. The first problem was figuring out how to get the molecular scissors inside the right cells without the body rejecting them. You can't just inject them and hope they find the right spot, because the body is a vast and complex environment. With 37 trillion cells, finding the right target is statistically worse than trying to win the lottery every day for years. The second problem was accuracy. What if the scissors cut the wrong spot and damaged a healthy part of the DNA? If you cut a tumor suppressor gene by accident, you might cause cancer while trying to cure a different disease entirely. For years, these problems seemed unsolvable, but then a breakthrough occurred that allowed scientists to bypass the delivery problem completely. For blood diseases like sickle cell, scientists realized they didn't need to fix the cells while they were inside the body. They could take the cells out, edit them in a lab, and then put them back into the patient safely. Think of it like updating software on your computer. You download the patch and install it to fix the system bugs. You do not update it while it is running critical programs. You take it offline first to ensure the process is safe. This approach meant they could control everything, performing precision editing in a petri dish where they could monitor every single variable. By 2019, they were ready, and the first human trials began with patients who were willing to risk everything for a cure. Victoria Gray raised her hand and said she would go first, stepping into the spotlight as a pioneer for genetic medicine. She had nothing to lose. With a lifetime of pain behind her and unknown risks ahead, she chose to move forward. But the hope of being the first person cured was worth any risk, so she prepared herself for the historic treatment. Here is exactly what happened to her cells during the process that would rewrite her genetic code and save her life. Step 1 involved doctors collecting her blood stem cells, which are the mother cells that create all other blood cells. Step 2 happened in the lab, where billions of these isolated cells received the CRISPR editing to correct their genetic instructions. The scissors did not fix the sickle cell gene directly. Instead, they did something much cleverer to solve the biological problem. You actually have a backup gene, one that makes fetal hemoglobin, which is usually hidden deep inside your genetic code. When you were in the womb, this gene made your blood cells, ensuring you had perfect, healthy cells during your development. But around birth, a genetic switch flips, and this gene turns off forever, forcing the body to use adult hemoglobin instead. The adult hemoglobin gene takes over, and for Victoria, this included the broken sickle cell version that caused her disease. The scientists' brilliant idea was to cut the switch, forcing the body to turn the fetal gene back on permanently. Suddenly, Victoria's body started making fetal hemoglobin again, producing healthy blood for the first time in 33 painful years. The healthy hemoglobin prevented the sickling, effectively neutralizing the disease, even though the original mutation was technically still present in her DNA. Step 3 was difficult. Before the edited cells could go back, Victoria needed intense chemotherapy to prepare her body for them. This destroyed her existing bone marrow to make room for the new cells, a necessary but physically demanding part of the process. It is like demolishing an old building to build a better one. You must clear the site before construction can begin. Step 4 was the infusion, where the edited cells were put back into her bloodstream to find their way home. It was like seeds being planted, and now they just had to wait and see if they would actually grow. The first month was terrifying because Victoria was vulnerable. Her immune system was gone, and she had to stay in isolation. Then, the signs of success appeared as her blood count started rising, proving the treatment was taking hold in her marrow. The edited stem cells were working, making new, healthy blood that would carry oxygen without turning into painful sickle shapes. 2 months later, Victoria left the hospital, stepping into the sunshine while waiting to see what her new life would bring. The moment of truth arrived. Would she have another pain crisis, or was the disease truly gone from her body? 1 month passed with nothing, then 2 months passed, and she realized she was living without the pain she always knew. 6 months became a year, and for the first time in her adult life, she was living completely without pain. She could take her kids to the park, plan vacations, and dream about a future that was not defined by illness. She enjoyed things most people take for granted, like tying shoes or carrying groceries, simple joys she never thought she'd have. Now, 3 years later, Victoria has had 0 pain crises, a triumphant milestone that confirms the treatment was a total success. Tests showed her fetal hemoglobin levels reached over 40%. In comparison, a normal adult typically has levels that are less than 1%. She is not just treated, she is effectively cured, thanks to a one-time treatment that rewrote her genetic destiny. This wasn't science fiction. This was December 2023, and it is happening right now, marking a new era in human health. Victoria's success wasn't just a personal miracle, it was proof of concept for the future of medicine on a global scale. Because the FDA did something historic in December 2023, validating the work of thousands of scientists over many years. They approved the first CRISPR gene therapy for general use, marking the first time such a medicine has ever been authorized. The drug is called Casgevy, and it is now available to patients who have been waiting for a cure for decades. But here is where we need to talk about the catch, because there is always a catch with new technology. The official price tag for this groundbreaking treatment is a staggering $2.2 million per patient, a number that shocks the entire medical world. That is the price of a house, and in most cities, it is the price of multiple houses combined. How can anyone afford that? Well, here is the surprising math that explains why this price might actually be justified. Treating sickle cell disease the old way costs about $1.7 million over a lifetime due to hospital stays and medications. Plus, there is the cost of missed work, lost productivity, and the tragic reality of an early death for patients. One treatment costs $2.2 million versus a lifetime of suffering and expense that often exceeds that amount in the long run. Insurance companies are actually starting to cover it because it saves money long-term, making the cure accessible to more people. But the real story isn't sickle cell. It is what comes next, as this technology opens the door to many cures. Because CRISPR isn't just for blood diseases, scientists are already working on using it to treat conditions affecting other organs. They are targeting muscular dystrophy, aiming to restore muscle function in children who currently cannot walk or move freely. Trials for hereditary blindness are already showing people regaining sight, seeing their loved ones clearly for the first time ever. They are looking at cystic fibrosis, heart disease, and even some cancers, expanding the potential of this technology every day. For HIV, scientists have already removed the virus from human cells using CRISPR, offering hope for a permanent cure. The question isn't if CRISPR will transform medicine, it is how fast it will happen and who will get access. But with great power comes great responsibility and great danger, which we must address before the technology advances too far. Because CRISPR doesn't just fix diseases, it can change anything in your DNA, effectively allowing us to rewrite human nature. It could be used to change height, intelligence, athletic ability, or eye color, leading to a future of designer humans. And in 2018, someone crossed the line, creating a controversy that shook the scientific community to its very core. A scientist in China did something the world wasn't ready for, performing an experiment that violated international ethical standards. And it changed everything about how we think about genetic engineering, forcing the world to confront the dangers of this power. His name was He Jiankui, and he shocked the world by secretly editing human embryos, crossing an ethical line that science respected. Those were not embryos for research. They became twin baby girls born in 2018, the first genetically modified humans in history. They were created without proper safety testing or consent, leading to a global outcry against the reckless use of technology. The world reacted with outrage, and he was sentenced to 3 years in prison for his illegal medical practices. But the girls exist, and they are growing up with genetic changes we don't fully understand, living as an experiment. This raised the question everyone was afraid to ask. Where is the line between curing disease and designing people? Curing sickle cell is clearly good, and making designer babies is clearly wrong, but the middle ground is very blurry. What about preventing Alzheimer's before birth, or editing out cancer risk? These are the gray areas we must navigate. What about making your child taller, smarter, or stronger? These enhancement questions are becoming more relevant as the technology improves. These questions don't have easy answers, but we need to be asking them now before the technology makes the decision for us. Because the technology isn't waiting for us to figure out the ethics, it is racing ahead at breakneck speed. Every month, CRISPR gets more precise, more powerful, and more accessible, bringing us closer to a world of genetic modification. What costs millions now might cost thousands in 10 years, and maybe even hundreds in 20, democratizing access to gene editing. So here is where we are, standing at the edge of a new era in medicine that will define our future. We can cure diseases that have tortured humanity for millennia, ending suffering for millions of people around the globe. We can give people like Victoria their lives back, allowing them to dream of a future free from chronic pain. But we have to be careful, because the same tool that heals can also harm if we use it irresponsibly. The future of human genetics is being written right now, and if this video helped you understand it, please subscribe today.